Okay, welcome back to the Classics of Immunology Journal Club. And before I start, I just wanted to remind you to like the video if you like it, and also click on the bell to get notified for more. And then also visit my website and check out the writing tab, because all of the classic papers in immunology are listed there. Before I get into the paper that I'm going to do today, I wanted to just alert everybody to the fact, for the B cell mavens that are out there, the time period that we're right into right now, which is sort of the mid to late 70s. There are several papers by Susumu Tonegawa, who cracked the problem of solved the problem of how can you have such specificity for antibodies and yet have such broad diversity. And so he did the critical experiments where he compared plasmacytoma cells and at the genetic level with mouse embryo cells, and showed that based on the restriction fragments that he had on gels, that there had to be two different gene sections in the germline that were separated by, lots and lots of kilobases and so forth. But then somehow they were brought together in the differentiated B cell plasma cell. So those were really groundbreaking experiments. I'm not going to review them here, but if you're a B cell maven and you're interested, there are several papers by Tonegawa listed there, and you need to look at the years, and also a couple of papers by Phil Leader, because everybody jumped on it after Tonegawa had his original paper that was published in PNAS in 1976, I think. And then Tonegawa won the Nobel Prize in 1987 as a single investigator, which is unusual because there's so many people working nowadays and everybody knows what the questions are. The problem is getting the answer to the questions. And that's what we're all about in science these days. So there's that, and I wanted to mention that before we start, because I really wanted to continue on the saga of the T cell story and the products of the T cells, just like immunoglobulins are the products of the B cells. The critical products of the T cells are not antibodies, which some people thought for a long time. That was the T cell antigen receptor was going to be a new class of immunoglobulins. Rather, the critical products of T cells are cytokines or lymphokines or interleukins. And so now today we're going to delve into to another one of my papers, this one from 1979 by Paul Baker, Steve Gillis, and Kendall Smith. It's entitled Monoclonal Cytolytic T Cell Lines. I was also taught to keep the titles as short as possible so people don't forget them. You get a long sentence, it's difficult for people to remember. So this is Monoclonal Cytolytic T Cell Lines, and it was published in the Journal of Experimental Medicine in January of 1979. And that was really critical in terms of what happened next after this whole thing. And to give you some background, Paul Baker, who was the lead author on this paper, it was my first postdoc. He came from a really good friend of mine that I'd met in France when I was there with Georges Maté by the name of Torgny Fredrickson. And Torg was an interesting guy. He was a hematopathologist and a mouse hematopathologist. His expertise was essentially the pathologic microscopy of the blood system in the mouse. And he was an expert in mouse leukemias and lymphomas and so forth. He was on sabbatical in Village Juif, where I was with Professor Maté at the same time that I was there. And we started talking, and ultimately it was over coffee in the morning. And then that led to lunches, longer lunches, and started trading scientific stories and other kinds of things. And he was great. Torg was like a novelist. He would watch all the French people with their characteristic body language and mannerisms, and also the way they carried themselves. And he would make fun of them. And so we were two expats making fun of the people in the surrounding. Well, and when I came back from France, then a couple of 3 years later, Torg referred one of his graduate students, Paul Baker, to do his first postdoc with me. That was good. It turns out in the introduction of this paper, that one of the first things we say is that, you know, people have, it's really sort of a hot topic right now to see whether or not you could generate T cell clones. Everybody was really, at that point in time, 1979, after Cesar Milstein and George Kohler published about monoclonal antibodies and monoclonal B cells, the natural thing to say was, well, let's get monoclonal T cells. Then we could do all kinds of things. We could find out what the T cell antigen receptor is at the molecular level. We could find out how they kill other cells at the molecular level. We could find out new differentiation markers on T cells and so forth. And so we go through all that in the introduction. We also then restate what we talked about last week in terms of the last two papers that we had reviewed, the CTLL-2, cytotoxic T lymphocyte line 2. Had at this point in time when we did this paper, had been in culture for more than 2 years, maintained its cytotoxic specificity of being able to kill allogeneic cells, allogeneic leukemia cells, as well as syngeneic leukemia cells, but did not kill syngeneic normal cells. And we sort of figured that, well, we probably have at least two different subpopulations of cells, T cells in this cell line, and some with specificity to allogeneic leukemic cells and some with specificity for syngeneic leukemia cells. But we didn't know that. We just assumed that, and we had to basically prove that. So we decided to see if we could clone CTLL2. And at the time, in the materials and methods in this paper, there were two basic fundamental ways that you could clone cells. The tried and true oldest method was to use soft agar. So you would mix up your cells into the soft agar at a little bit elevated temperature so that the soft agar was liquid. And then you would plate those very dilute cultures of cells out in Petri dishes and then put them in the incubator at 37 degrees and hope that they would grow colonies. And so in a colony, as defined at that time, was all the progeny of a single cell. So that was a clone. And people had used this technique to clone tumor cells and all kinds of things. Nobody really had long-term T cell lines or long-term normal cells that they could try to clone. And so when we found that we could use this conditioned medium, T cell PHA stimulated conditioned medium to grow cells in liquid culture, we said to ourselves, well, this is like, ought to be like falling off a log. The other method would be then to do it in what they called limiting dilution. So in that particular instance, what you would do is you would dilute out your cell populations from your flask or wherever it was growing into a very low density of cells, and then plate them out in microtiter plates. And so in order to do this, we used as our growth factor the 48-hour Con-A T cell soup. And we took our CTLL2 cells that was growing in 1-to-1 TCGF and that had been growing in for months this way, retaining the cytotoxic specificity and retaining the theta antigen on the cell surface that we tested periodically. And also they remained surface immunoglobulin negative, so they didn't suddenly become B cells, which is good. And so we cloned these cells by limiting dilution. And the way we did that was that we used an electronic cell counter, a Coulter counter, and we would take the cells out of the incubator and count them. And based on what that count was, we would make an initial dilution of something that was at a reasonable density. So like, 2, 3, 4 million cells per ml. Then we would recount that that once we made the dilution, we were very careful in terms of we would recount those cells, 3 or 4 times, making sure that the density was what we knew it was. And then we would make tenfold dilutions from that until we got down to about below 3 cells per ml. And then we would then seed those cells into microtutter plates at 100 microliters per well, or 0.1 ml. So if we had 3 cells per ml, then we would get down to a density of 0.3 cells per well. Now, at that concentration, if you then did the Poisson statistics, you could then convince yourself and the statisticians anyway, that the probability that there would be more than one cell landing in a well was very rare or statistically significant at that stage of the game. So that's how we did our cloning, and I'll get into the results in a second. What we found that the soft auger thing was not a good way to clone. And limiting dilution was the way to go if you had a T cell growth factor. So in the results section of this paper, and I forgot to say that I am going to do now a screen sharing. So this is figure 1 from our paper. And as it says in the caption here, this is a cytotoxicity against our starting cells, our starting cell line, CTLL2, against allogeneic targets versus syngeneic targets, tumor cells, versus normal syngeneic cells. And this is the effector to target cell ratio. As we increase the ratio of killer cells to target cells, we get increased killing. And this is the percent cytotoxicity on the X or the Y-axis. And you can see we got more cell killing with the allogeneic targets than we did with the syngeneic targets. And so that's why we assumed that we were seeing at least 2 different subpopulations in this whole process. So we cloned the cells by limiting dilutions. And this is table 1, shows the results from our clonal isolation that we did. And we've got over here on the left-hand side, the clone number. We got 24 clones out of this. And this is the number of cells plated per well from 0.3 down to 0.13. This is the Poisson probability that there's more than one cell in a well at this cell density. And it was less than 0.05. And down here it was 0.01. And this is also the expected plating efficiency when at this cell density. And we plated 24 wells, and we expected 6 positive. We got 6 positive. That's 100% cloning efficiency. And you can see at this cell density, we had 90% cloning efficiency. And at this cell density, we had 67 and 70% cloning efficiency. So that basically was unheard of. Nobody was, most of the time, particularly when you use soft auger cloning, you got a cloning efficiency of about 1%, 1 or 2%, if you were lucky. A lot of times you didn't get any clones at all. So that was good. So then the next thing we wanted to do is we tested all of those clones for cytotoxicity against allogeneic tumor cells and syngeneic tumor cells. And you can see that these are representative clones against the allogeneic tumor, which is in the closed circles, and the syngeneic tumor, which are the triangles. And this is clone 3, which has got allospecificity, but no syngeneic tumor specificity. And the reverse here is clone 15, and then clone 24 that kills both about equally, and then clone 20 here that didn't kill anything at all. And the thing is, of all the clones that we had, almost half of these were alloreactive. About 20% were syngeneic tumor cells. About 20% were both, and 10 or 15% were negative. And so 85% of the clones that we got out of our clone cell line were cytotoxic, which was also very heartening to find. And then we wanted to see how stable the cytotoxic specificity was over time. And so this is figure 3 of this paper. And the upper panel here are lysis of the syngeneic FBL3 versus the allogeneic tumor cell. And this is clone 15, which was our syngeneic specificity at 4 weeks and at 8 weeks and at 13 weeks of culture. And then this is clone 20 down here, which had no cytolytic reactivity at all. And even up to 1,000 to 1 ratio, we didn't kill normal cells, either thymocytes or blasts. So that was good. And then the last figure here, sort of like the pièce de la résistance, is that we wanted to see whether or not, if the clones were really clones, that is the asexual progeny of a single cell, then if we cloned the clones, all of the subclones should be identical. And you can see here in A, B, C, D, and E, and F, there are 6 clones out of this from clone 15, and they were all had the same basic specificity of their antigen reactivity of cytolysis. So that was it from the standpoint of the data in this paper. And we were really happy about this. We thought we'd done all we possibly could do to prove, because that the critical question is, are these really clones that you think you have, Smith? And of course, it would be good if they were. So the discussion of this paper is, we make the following points. The first is, this is the first report of cytotoxic T lymphocyte clones. Nobody else has done this before. Secondly, if you use these methods, you can generate as many clones as you want. You can get more clones. And in particular, because we'd all, at this time and at this point in time, we'd already made human cytolytic long-term T cell lines growing in T cell growth factor. Then we said, well, okay, well, now we can get human T cell clones. That's very important, and I'll get into that in a second. The fact is that 80, as I've already said, 80% of these clones from CTLL2 had cytolytic reactivity. And we speculated in the discussion about whether or not this meant this was because the cytotoxic T cells were more reactive to the T cell growth factor than T helper cells or T suppressor cells. There was still this phenomenon of T suppressor cells that was floating around, particularly in the 1970s. We didn't know that might be the case, but we wondered about that. And there'll be more data coming down the line about that particular point. But finally, what we said is that now it's possible to study a T cell mediated cytolysis at the molecular level. That was the paper. That's what we said in the paper. We fired this paper off this is the subsequent happenings for this particular paper. We fired it off to Nature again. They'd taken our first paper finally, and we got it rejected. In return, they never sent it out for review. But this time I said to myself, these people in Nature just are not very bright. And so I just said, we didn't go back. I didn't try to educate them. We sent it directly off we just reformatted it and sent it off to the JEM, Journal of Experimental Medicine, which took it right away. And this was in 1978. It wasn't published until 1979, but it came out in January of 79. Now that's important because a lot of other people, maybe not a lot, but other people were trying to do this too. So the priority in this paper and the month as well as the year are very, very important. The result of this paper being published in January of 1979 was that I started getting invited to all kinds of meetings, and it was clear that we were no longer working in obscurity anymore. This put us on the immunological map, and me in particular, but also Paul Baker, and Steve Gillis. Everybody's on the from Dartmouth now. And Dartmouth was put onto the immunological map for the first time. And it's interesting, you know, of our so-called competitors, one of them was Bill Paul's lab at the NIH. And when I was invited to go down there and talk to them and so forth, and in just sitting down on a one-to-one with Bill in his office, he said to me, he says, well, you know, we don't really believe that you really have proven that you've got T cell clones. And I looked at him and I said, what do you don't believe in mathematics? But anyway, we felt we had, everybody else in the world felt we had, and so that people were off and running. It created a huge uproar in the immunology. Everybody wanted to make their own T cell clones to their favorite antigen, whatever it was. And one of the most favorite antigens in immunology, or molecules, was the T cell antigen receptor. And as we predicted in this paper, this ought to be what you ought to be able to do is make antigen-specific T cell clones, immunize mice with these T cell clones, and then screen for reactivity that would be clone-specific and wouldn't recognize all T cells and so forth. And of course, because the immune response is not only antigen specific, it's species specific. And so the way to do that experiment is to make human T cell clones and immunize mice. And that turns out exactly what Alice Reinhardt's did a few years later. Meanwhile, most of the immunologists, I would say 99.9% of them, were trying to do this by making mouse T cell clones and immunizing mice. And that was significantly more difficult. And they got beat by Alice. And so I'm going to stop there. And so if you like this video and you think this is interesting, an interesting exercise, like it on the YouTube channel. Also click on the little bell to get notified. Scroll down and look up these papers and read them yourself. Thanks. It's been fun.